I'm going to introduce to you Dr. Ana Maria Vekic. She is a graduate in chemistry, and she's also a teacher in science, and she did that with the Catholic University of this Rio Grande do Sul. She also has applied technology to phytosanitary measures, and she has a specialization in technological development for industries by the National Center of Research and Biotechnology, and now she's working at Anvisa, the National Agency for Sanitary Surveillance. Thank you very much for coming.
Thank you for the organization of the event. Good afternoon to all. What I'm going to try to bring today, Luis Claudio has already said a bit of how Anvisa is organized, and he said that it is divided by products, but not processes. In our area, that is toxicology, where we see especially the agrotoxics and also other substances that have no shelter in other areas that were visited this morning. So toxicology ends up by we're going to put our experience with agrotoxics and see what is happening with nanotechnologies. As was said this morning, it is very important that we see that we have a backup, and this will be with the laws and norms that can make us regulate it. It is interested that we see what is interesting for society. To authorize is very easy, but the effects of the authorization will be seen only 20 years ahead. And if you do not authorize, we don't have solid arguments to do this. This has to be based on science and instruments that will permit us to make these decisions. So it is very, the regulatory backup is very important. When we talk about the agrotoxical laws, Brazil since 1934, Brazil had a regulation for agrotoxics, and it was the one that prevailed until 1939. Until 89, sorry. What happened between this time in the 70s, the discovery of the benefits or not of the agrotoxins, and this mobilized society, and the health ministry begins to be part of the assessment of the agrotoxics, and this began to happen. The Minister of Health started to assess this in the 90s onwards, and only in 1989 the legislation have become to involve this. From 1934 until 1989, we needed all these years to discover something to be able to regulate it. So what society expects of us is that this experience doesn't have to repeat itself in such a long and distant future. So it is important that we can foresee and think this situation for it not to happen again with what happened to agrotoxics. So here we have the law of agrotoxics, the creation of the system of health in Brazil, the SUS, and also of publicity, the law of publicity, and and the regulations of three or agencies that are health, environment, and agriculture. So I'm going to make a parallel, and the assessment of these products are between these three. We give the register, and when this product is already registered, and we need a feedback to see if the assessment of this product, especially for health, and it is reassessed, and this register will. And this has a benefit because we will not have to revise each 5 years, consuming our workforce to revise what is not important, but centralizing and focusing on what is important to review this data. So this is part of the register of the national product. Now I'm going to see in what has to do with the toxicological assessment. So this has to do with the three agencies. So let's see exactly this. What are this assessment? These little mice are important for us to make the decision. There's much that there are many guidelines that cannot dispense the use of animals. When we talk about regulation, we're thinking of assessing toxicologically this product. The category of products is one that everyone is exposed to independently of our will. In one way or another, we are going to be exposed to them, and we have to be concerned about it. As occupational exposition, as for example, the worker who is going to be applying this product, and people who are going to work with nanomaterials, and the population as from child to the elderly, and even the child before it is born, when it is in the uterus. So with this, we have to work with guidelines and very clear methodologies. These methodologies have to be accepted for regulation. They have to be internationally accepted. If they are like that, we will be able to compare them. We can have a reproducibility. We can assess the relevant endpoints, and we'll have the uniformity of treatment. How will I define the assessment? We'll have to have a uniformity in the treatment. So this is very important, that this is harmonized internationally. These guidelines, there are very known guidelines that are known from the United States, from Australia, from Japan, and they are being assembled in the last years. But inside the CCE and all these countries that have methodologies are part of it. They are uniformed treatment. Our protocols have to be validated and accepted so that they can be used in regulation. With all this, we need to work with methodologies that don't have a defined guideline. When we do not know, the guideline has to be discussed with the regulatory agencies, because if they don't work with a harmonized guideline, they're not going to work with the good practices in laboratories with these principles. So it has to be agreed with the regulatory agencies before the study. All this permits us to work with good practices in laboratory, and this is where the INMETRO enters. It assesses these studies, and the onus of this test is who wants to commercialize this product. The industry has to approve its own product, that it is safe. So we have to work with good practices in laboratories, but this is going to be able so that we can track the results. With these guidelines, we're going to start an assessment, and we're going to begin an assessment, and we're going to begin with the process of the synthesis. We're going to discuss the impurities. We're going to make analysis of quantitative and qualitative analysis of 5 batches, and we're going to characterize the products, the physical chemical studies and the toxocinetic studies. We start making a parameter from this data. I'm going to make a parallel with nanotechnology. What we know here and what I'm bringing here is part of a group of work that was made inside the CTA that establishes minimum data for some products. This is what we're going to see here, the comparing agrotoxics with nanoproducts. So this is very similar to the agrotoxic identification of products. So we see the composition of the nanomaterial, including the degree of purity and impurity known, and the additives, the basic morphology, description of the chemical surface, and it is a little bit different of what we have in the agrotoxic. Here we see the physical and chemical properties. When we talk about those of agrotoxics, this is where begins the big difference between the products that are made of nanoparticles and the agrotoxical products. Here we see an identification, and how am I going to reproduce this identification in laboratory? And this is where the differences start. We'll see this in the toxocinetic of the product. We're going to see the absorption, distribution, and the excretion. We're going to see how these products are distributed in our body, our organs that are targets, and this we will have to study between how this is going to be distributed in our body between the absorption and its excretion, and how the metabolites are going to be studied, and how when we think in this study, it is one of the principal difficulties that we have for nanomaterials. Why? Because the method has to be determined for chemical substances. We're going to mark them radiologically, but how are we going to mark them with the nanoparticles? It can change its behavior from one state to another. So this is some of the studies that we will have more difficulty in the methodology in the follow-up of the methodology. We're going to see some studies that have a restriction of registers, and we also have the mutation studies, and we also have the classification and the assessment of danger. And all these studies, I'm quickly showing these methodologies that are followed in the CT, all of them are the same that are utilized and the same that are being indicated to test these materials. After toxicity is determined and established, we're going to depend what is the form of the material. And so these studies that are being recommended in the acute point of view or in the chronic point of view are the same methodologies. Some are targets or gates, so that we have to be more careful. A possibility of phototoxicity, but all have the same methodology that at least are the ones that are followed. Here we have the toxicological classification and the point of view. And when we talk about reproduction, all the toxicologies, not all are the same. Like for example, when we talk about inhaling toxicity, we have to explore a bit more. But it is basically the same methodology. There are some studies that are for longer time. They represent a cutoff. They prohibit this product to enter society. So we establish here a toxicological assessment. All of them, the residual studies with a formulated product, and after this we have the assessment of the diet risk, that until you reach to the maximum limit of risk. Now I'm going to talk about of all these methodologies that were recommended in this group that were formed in the OECD in 2004 to establish what existed in nanotechnology, what existed in regulations, and what was being done. In 2006, they established a working group, which they made 6-7 subgroups, and they had as an objective, they wanted to maximize the environmental benefits and minimize the impacts of nanotechnologies and the products that had nanotechnologies. And now there is a manual in June of 2010. We have a first version of this manual, and the tendency is that it will be modified. And they established 14 nano products that are being commercialized or that are being commercialized to be studied, that should be studied based in all these methodologies that I talked about. So after the assessment of these 14 products, we will have more scientific information for us to have to be assessed, and this should happen in the next 10 years, and this has to be defined and assessed in this period. Well, I don't know if these are conclusions, but reflections of what is happening, and up to now we have different kinds of nanomaterials, and for them we will have different methodologies of analysis. To define this as a rule, we have to think of in each kind of product how the intention of this product. We have to know the knowledge of the applicability and which methodologies are being indicated, but they will also be used for the first time for those products. So we have to think of the finality of the products, and there are no specific rules in which we are working in this path. It is really research. There is a necessity of regulation, but how should this regulatory approach be? Everything has to be regulated. We have to know what is being produced. Some products maybe don't need a specific regulation because will this be sufficient for a decision making? But we have to define where these substances are going to be included, and of course for this decision making, the scientific evolution in the production must be accompanied by the assessment and of the evolution of risks for the health of the workers and the population and of the environment. So we have to with agrotoxins, they took a long time to create the law that only happened before the Rio 92 convention, 92 convention. So we wouldn't want this to repeat with this category of products. Thank you very much.